This is the second video of the Control of Gene Expression for Neuroscience lecture. In this video, we will talk about techniques to measure transcription. Within the outline of the entire lecture, as I mentioned earlier, we will talk about techniques to measure transcript, specifically focusing on the reverse transcription and cDNA synthesis. This technique is called reverse transcriptase-quantitative polymerase chain reaction, or commonly known RT-qPCR. RT-qPCR consists of cycles of repeated heating and cooling, melting and enzymatic amplification of a complementary DNA molecule, or cDNA, that is synthesized from messenger RNA. RT-qPCR is used to quantitatively detect gene expression. To perform qRT-PCR, we need a couple of different elements. We first need to synthesize cDNA. This consists in the transformation of messenger RNA into cDNA. The second element are the primers. Primers are required to enable selective and repeated amplification of the cDNA. And third, we need to perform the quantitative PCR. This consists in the amplification step to quantitatively measure the amount of a specific cDNA. qPCR is performed by the use of a tag DNA polymerase that will amplify the cDNA, PCR reagents such as the NTPs, magnesium, and a tag buffer, and then a fluorescent dye that is necessary to detect the DNA that is amplified, and it will be used to quantitatively analyze the amount of cDNA that is amplified. So to start the RT-qPCR, we first need to synthesize cDNA. To synthesize cDNA, we need different elements that require the use of a reverse transcriptase that has the ability to copy RNA into DNA. We will use also an oligo-DT primer that will hybridize to the poly-A tail of the messenger RNA, and also the NTPs. The reverse transcriptase will copy all the messenger RNAs into a single-strand cDNA in this first strand cDNA synthesis step. After this first step, then we will use an RNase H that is required to degrade all the RNA template. Then once we have degraded the RNA, and then we have a single-strand cDNA, we will use a DNA polymerase plus the NTPs that we will copy this into a double-strand cDNA. After this process, we have all RNAs transform into cDNAs that can be used in the quantitative PCR. Then, the second step is primer design for RT-PCR. We need to design primers for a specific transcript of interest. So then that will let us amplify the cDNA of a specific gene of interest that we want to study the transcription. Here I put some guidelines on how you can access to the genomic sequence of a particular gene of interest, and also the software that you can use to design this type of primers. You will need to design a forward primer and a reverse primer to selectively amplify a cDNA of interest. These are general conditions that are necessary to design primers with good quality for qPCR. And if you want to know more about how you can design primers for RT-PCR, then you can visit this website that provides all the details necessary on how you can design primers for RT-PCR. So now we can actually perform the quantitative PCR. For that, we will start mixing the cDNA with the PCR reagents that we just mentioned earlier. We will also include the primers and the cyber green dye. The very first step of the qPCR is denaturing double-strand DNA into single-strand DNA. Now, the primers can anneal into the specific target, a specific cDNA. This process is called annealing. Now, the DNA polymerase can start copying this single-strand cDNA into double-strand DNA. This process is called elongation. During the elongation step, the cyber dye gets inserted into the newly generated double-strand DNA and starts to fluorescence. Then, we denature this newly synthesized DNA to go back to single-strand DNA and start a new PCR cycle. The aim of this is to amplify the DNA and increase the fluorescence signal. Then this PCR reaction finishes approximately after 35 to 40 cycles, and a final elongation step is necessary to finish copying all the double-strand DNA molecules. During this PCR reaction, we can monitor how the fluorescence signal increases in real time, and we will use this signal to determine how much cDNA of our transcript of interest there was in the sample. Typically, an amplification curve has 3 phases. The first is called the initiation phase, and it occurs usually during the first PCR cycles when the emitted fluorescence cannot be distinguished from the baseline. Then there is the exponential phase. This happens when there is an exponential increase in fluorescence due to an exponential increase in DNA. And then there is the plateau phase, which is reached when the reagents of the PCR are exhausted. The fluorescence emitted in these very first steps of the PCR are used to calculate the baseline. Now, to quantify the amount of cDNA that we had, we use the CP, or crossing point PCR cycle, which is actually the PCR cycle at which the fluorescence achieves a threshold above the baseline. The CP corresponds to the cycle at which a statistically significant increase in fluorescence is first detected. The number of cycles needed for the amplification-associated fluorescence to reach a specific threshold level of detection is inversely correlated to the amount of nucleic acid that was in the original sample. This means that the sooner we see signal coming, the more cDNA there was for a particular transcript of interest. So how do we analyze the data generated from the qPCR? Well, as I just said, we will be using the CP values calculated from the amplification curves. Just one note, some people uses the CT values, which are threshold cycles, and they're basically just the same as the CP values, okay? They're just like 2 different ways of naming the same type of values. CT means the same as CP. And there are different methods to analyze qPCR data. Here I just describe one of them, which is the delta-delta CT method, which is the very first method that was used to analyze qPCR values, and it is still used. There are now different methods more complicated than this one. They use different algorithms to interpret qPCR data, but we will focus on describing the delta-delta CT method. And this method is really very convenient to analyze relative changes in gene expression. It requires the assignment of 1 or more housekeeping genes, which are assumed to be uniformly and constantly expressed in all samples. And these are just few examples of housekeeping genes you can analyze. You have to determine experimentally which ones are better for your conditions, because sometimes ones are not very good for your experiments. And you will also need at least to determine 1 control sample. So then the expression of the experimental samples will be compared to the control sample, and that's why the analysis will show relative chain expression. So what you do is you take the average of the CT values for the housekeeping gene and the gene being tested in the experimental and control conditions, and this will give you 4 different values. The 4 values are gene being tested in the experimental condition, or TE, the housekeeping gene in the experimental condition, HE, the gene being tested in the control condition, TC, and the housekeeping in the control condition. And now you calculate the differences between the TE and the HE, these 2 values, and the TC and HC. And these are your delta CT values for the experimental sample and the control sample. Then you have to calculate the difference between delta CT experimental and delta CT control. And this is what gives you the delta CT. And since all calculations are in logarithm base 2, every time there is twice as much DNA, your CT values decrease by 1. So then you have to use this formula to calculate the fold change expression in your experimental sample compared to the control. In this case, there is a fold change of 0.6, which means that the gene of interest is downregulated in your experimental conditions compared to control. If the values were higher than 1, then you could start considering those being upregulated. And we will do some problems at the end of the video to practice how to calculate the delta CT method, and also in class we can practice more on this issue. So this is an example on how to apply RT-qPCR for the profiling of single neuronal cells in the context of lineage-specific reprogrammed human neuronal cells. What the authors did was isolating single neurons expressing GFP as a marker for reprogramming and doing RNA extraction from every one of these single neurons. The authors also used a specific dynamic array that let them use high throughput expression profiling from single neuronal cells, assaying up to 96 independent samples with up to 96 different qPCR primers in one single experiment. And this is the data analysis shown on the paper, and on the side you can find some of the genes that they tested in this array. At the top there are some of the housekeeping genes used, CAPDH and tubulin, which is another cytoskeleton gene. And every column here in this diagram represents in single neuronal cell and the profiling of all these genes. And really, single cell gene profiling can provide definitive evidence regarding the conversion of one cell type into another at a really high level of resolution. And if you want to know more about this technique and how the authors applied RT-PCR into the research, you can find more information in these 2 references in Nature. So finally, what are the advantages and disadvantages of using qRT-PCR? Well, on the con side, really you need to have hybridization conditions that must be similar across different probes or primers. If you're targeting different RNAs, they really need to have similar conditions on how they amplify the targets, so then you can compare different genes with each other. And obviously the structures are destroyed, so then you lose cell context. But other than that, this technique it's really specific and very sensitive, and is definitely user-friendly with high quality control, and it's accessible by many labs, so you can do qPCR in pretty much any lab where you want to determine the transcription changes in different samples for a certain amount of genes. Here there is a problem where you can practice some of the things that we've just seen in this video. This problem is about determining whether the expression of synapsin 1 changes during development in the cerebellum. And there are different samples you are prone to analyze, and the first step of the problem is describe all the steps necessary to analyze the expression of synapsin 1. Then go through the first slides and see what do we need to really analyze the expression of this gene. Then the second part is calculate the fold change expression in the different experimental samples. And I'm just telling you to consider E10 as your control sample, and GAPDH will be used as your reference or housekeeping gene. And then the last question is basically interpret the data that you obtain from question B, so then you can describe whether the gene is downregulated or is upregulated, or is it not changing during development. We will see this problem also in class, but I encourage you to do it at home first, so then we can go over all the things that you didn't understand.